Right. Okay, there is a call.
Team Prithvi, can we start?
Yes, we can.
Good morning and welcome to yet another session of Chai and Why. And we are very happy to be back at the Prithvi Theatre in Mumbai. Before we start today's session, just in case people are joining us for the first time, Chai and Why happens on the first, third, and fifth Sundays of the month at 11 o'clock. At first Sundays, we are always at Prithvi Theatre, and starting this year, we are back again with our physical sessions. Our third and fifth Sunday sessions are still online only since our college and school partners are still not allowed to open up in Mumbai. Once they open up, we'll be there as well, but we will also be doing these online webinars and live sessions on YouTube and Facebook as well. Now, remember that if you're watching us online, there is no way for us to get feedback from you unless you type in your questions on the question and answer box if you're on Zoom, or in the chats and comments on YouTube and Facebook. So please do give us your comments. Please do ask your questions, and we will be very happy to take them during this session. Now, of course, before we get to today's topic, I'm sure we are all really interested to know about vaccines and COVID. That is the most important one of the topics right now. Let me also take a few minutes to tell you the upcoming programs. I will make a formal announcement at the end as well, but just remember 2 weeks from now, we will be going back to one of our live from the lab sessions where we're going to be up close with scientists in the lab. We will be at TIFR looking at how materials are sort of investigated using light, something called optical spectroscopy. We will be looking at that 2 weeks from now. And a month from now, March 7th at Prithvi, remember March 8th is Women's Day. And on March 7th, we're going to have a very special session looking at many of the forgotten heroes of science who were women who just didn't get recognition. And we will be having a special session on that on March 7th. Professor Vandana Nanal will be doing this at Prithvi. So those are the sessions coming up. But also remember, February 28th is National Science Day. We will also have some sessions. Unfortunately, we can't have the huge science mela that we usually have. We will be having the online versions of Meet the Scientists, do some experiments, see some labs. All that's going to happen online. I will give you the details at the end of this today's session as well. But right now, it's time for me to introduce Professor Sandhya Koushika, our speaker for today. Sandhya is a biologist at TIFR whose normal research interests is in tiny creatures, worms, in which she studies what happens to their neurons, inside their neurons, transport in their neurons, and everything on that. But Sandhya has also played a very crucial role during this COVID-19 pandemic, taking the lead in two respects. One was the COVID-Gyan platform, which is many institutions in India together, as well as a much more informal effort, the Indian scientists' response to COVID. And thanks to that, she's actually been doing a lot of public engagement on COVID. And today she's going to tell us about vaccines. So with that very short introduction, I'm going to hand it over to Sandhya. So Sandhya, all yours. And just audience, please remember, let us know where you're watching from on YouTube and Facebook. We'd love to know where you're watching from. And do type in your questions and keep your comments. We'll try and get them as soon as we can. We do have a few volunteers who are monitoring the Facebook and the YouTube and the Zoom webinar question and answer channels. We'll be there with you very soon. So with that, I'm going to turn off my video and mute myself, and over to you.
Hello, everybody. Can you hear me? I'm going to assume that you can hear me.
Yeah, so people on YouTube, please comment if you cannot hear or if the sound is not good. Please let us know in the chat.
And people in Prithvi Theatre, am I loud enough? Yes, thank you.
Yes, I think we can hear you. So good to go ahead.
Okay. So I am an accidental COVID-19 science communicator, and it's been a wild ride. I don't think any field of biology in my lifetime has generated as much science. And I'm going to do my best to sort of share some of the things I have learned along the way. And hopefully, it will be an engaging session, and feel free to ask questions at any time. And, I'm sure Arnab and others will moderate the session. So today's topic, which Arnab wanted me to talk about, is vaccines. And 11 months ago, March of last year, last COVID, last Jan-Y that I gave was end of March last year, was talking about when just the pandemic was unfolding. So while all of you are looking at this, see how far we have come and how swiftly we have come. And I like to think, like many others, that science has played a very crucial part in making sure that we are here today looking at a way to protect all humanity from this virus. Okay, but let's look at the past. And if I can only, I'm going to start with this. I should turn on the laser pointer. I'm going to start with this article which struck my mind, which came out in BBC. Diseases have been around, infectious diseases have been around since time immemorial. And amongst all the very many diseases that are there across the world, one we can be quite sure has been eradicated is smallpox. And it was eradicated by concerted efforts, which took a very, very long time. And I will show you some data about it. But this is a picture, a painting which one of the historians saw when it had come up for sale in Sotheby's, and suggests that this is a picture which sort of highlights the fact that the queen of Mysore, of the kingdom of Mysore, had actually vaccinated herself. This way of holding up her sleeve and showing her arm is supposed to indicate that. Right next to her is her mother-in-law, who lost her husband to smallpox and was very keen that her son and daughters get vaccinated. So at once, and we're talking about 1800s, at once this is a picture which illustrates how you communicate, how first people who are in positions of power vaccinate themselves, share that information with the public to build confidence, but it's also an example of how early vaccination started. What is not clear from this picture is whether it was vaccination or variolation. And I'll just come to it in the next couple of slides, which are very different approaches. Smallpox has been known since ancient times. It is believed to have originated perhaps in India, perhaps in Egypt, but certainly in this part of the world has played a part of it. And as early as 300 BC, you see description of smallpox in Sanskrit literature. What was known since then is inoculation was reportedly practiced, and this information of how this inoculation was practiced was shared in London. So that information flow was taking place where people were desperate to protect themselves and their populations from this horrific disease. The way this vaccination happened is they would take dried pustules, grind them up, either inoculate them into the person or, push it up their nose, and you had very little control. So the people who were doing vaccinations, who were sometimes called tikadars in India, were very skilled practitioners. They knew how to titrate the dose, because if you got it wrong, you would have a very severe disease. There were very complex ways of doing this kind of variolation across the world. For instance, John Adams was treated with things like mercury before inoculation, and in fact had got to a situation where he thought he was going to die for all of these balancing treatments to protect him from having a severe disease. And lifelong he had teeth which didn't quite fit, because it had sort of loosened from his gums. So people had been trying a way to say, can we take some material from some person who has been infected, attenuate it or reduce it in some way, give it to someone else and give them a milder form of disease. Okay, but India has been up there, as you see, those practices were shared across the world in one form or the other. The first documented smallpox vaccination was done in 1802. And you can see the picture, the marriage between the people which I started the painting took place in 1805. Presumably this painting was still in the early part of the first decade of 1800s. Vaccination really came from Edward Jenner's. He noticed, he was a pretty big part of it, although there had been other studies around that time, showing that you could probably take people who had been infected. Milkmaids never got smallpox. They were probably getting infected with cowpox, which was giving you cross-reactivity to smallpox. And this is again going to be perhaps important even when you look at the coronavirus, vaccines against coronavirus. What is very clear from what I told you before is this inoculation or variolation means of producing an immune reaction in a human being to protect them is very dangerous. Actually, lots of things can go wrong and you can get very severe disease. So once vaccination was considered to be, something which was safer, people started banning inoculation. Now, this was a very, very sensitive issue because it was the Britishers who were trying to vaccinate Indians. And we had our own traditions of variolations and tikadars and people who took care of these issues for our community. There were worries that were these vaccines raised in cows? Were they raised in pigs, which sort of, came up against what were social practices of our time, right? So hand in hand, as vaccines have been evolved and been available to communities to protect themselves, side by side, there have been issues about, should we be taking it? Should we not? And all the things that come with hesitancy, along with political considerations. If you have vaccines delivered by doctors, all the tikadars are out of a job. Who is going to support them anymore? So these are all issues which are not to do with the science, but issues that surround a vaccine, some of which still plague us today. Okay. The initial resistance, as I said, comes due to various reasons, some of which I have discussed here today. And in fact, the government in the 70s made a very, very big push to sort of wipe out smallpox in the country. And here is a picture that I took from an online exhibition. And these were the kinds of posters where they said, please report right away. If you have symptoms, please report right away. And if some of you here who are in their 40s, speak to your own parents who are probably in their 80s and they came from more village-like environments, they would actually tell you that there were mechanisms of isolation in homes that showed positivity for smallpox. The same sort of public health measures that we follow today with COVID-19, saying you must isolate the people who are testing positive, right? Except in that case, in the past, when people isolated, it was because they exhibited the disease and you knew it to be an infectious disease. Oh, wow, that was ominous. So I just wanted to share a graph with you, and you will see this repeatedly in other examples. This is from a doctor and a scientist who worked on the ground. He was a Japanese, and this is an article which you are free to read. All of these articles, you will be able to find the links if you go to the YouTube video. And you see how patchy. You had some cases, it came down, it went back up close to 100,000. And the last major outbreak, you had almost 200,000 people having smallpox vaccine. The WHO, sometime in 1958, took a resolution that we have the means to get rid of smallpox. We should try to eradicate this virus. And it was possible to do so because there were no non-human hosts. It was restricted to human beings. And it was therefore fundamentally possible to eradicate this the way it may not be possible for many other kinds of diseases. In fact, one of the techniques used, and this was used in multiple places, but certainly in India in the late 70s, was you would see one case which was reported, and then people would doctors and their WHO team would go in and do what is considered a ring vaccination. They would vaccinate everybody around that individual, essentially a means to break the chain. It is something that we heard very early in the COVID-19 epidemic, saying we must break the chain, and the way to break the chain is to take all these public health measures which were, were been known to work or suspected to work at that time. So here you break the chain by immunizing people around the person who got infected, and that was called ring vaccination. So these are concepts that have been around, as you see, for a very long time. Deaths decrease with vaccination. Here are examples of smallpox in London, polio in United States, and measles. And you can look at the years. Here we're looking at 1630 to early 1900s. And this is percentage of all deaths. And at some point, 20%, close to 20% of the people were dying due to smallpox. You have the vaccine, cowpox-based vaccine, introduced, you have variolation banned, and when you have large gatherings of people like the Franco-Prussian War, where people are just fighting with each other, you have a spike again, and then it comes down. Same thing with polio, very good success in terms of polio. You don't have this jaggediness. And you look at measles, which is more recent in time. You have a large number of people who get who have it, about 4% of the population of all deaths in Nigeria occur from measles. Massive vaccination campaign comes down. There is Gavi Vaccine Alliance supports the vaccination effort and also maybe assists with trying to, make low-cost vaccines available, goes down again. But it's still not to zero, right? But never mind, there's a very, very big change once you have a national immunization campaign, but no eradication. Very different from smallpox, where some years ago we considered whether we should destroy the last bits of smallpox titers that were present at all. Okay, I just thought I would show this because someone not that long ago, I was giving a simpler version of this. The Tamil Nadu Science Forum said, I think the biggest public health measure which got rid of all these diseases was actually hygiene, just having good toilets and ability to wash hands. And I thought this is a great illustration. I know it's very hard to read that slide, but I'm really only asking you to look at the circles. And if you do want to, please grab the reference later and you can read it yourself. You can see the picture yourself. We are looking at the 90s. We're looking specifically at United States. The size of the circle is the number of reported cases. Okay? And just look at the size of the circle, and we're looking at the disease chickenpox. Chickenpox, you have a lot of cases being reported, 100,000 every year, just going on. 100,000 is not a small number. You now have the chickenpox vaccine. I think nobody, 1995, I was doing my PhD. It was definitely, I don't see any difference in hygiene practices between 1995 and now in terms of, first world countries or even in good situations in India. But in the US, certainly no difference. You introduce the vaccine, the number of cases really, really goes down. So while hygiene might have overall improved, there is still room for vaccines. In fact, vaccines should be available for all stages of life. And I'm pointing down specifically for chickenpox because you know that people who get infected from chickenpox at a young age can, as older people, get shingles because the reservoirs of this virus stay within your body. So vaccines are relevant for all ages and all stages, and public health improvements is just public hygiene alone do not mitigate this need for having protective measures against infectious diseases. So let's come to COVID. I think I've sort of given you a little bit of introduction up to vaccines and what we saw in India, especially with smallpox. I think we have all, one way or other, there's a tremendous amount of fatigue, but the numbers are staggering. I think none of us and no public health individual has ever seen numbers like this within such a short period of time. Let's just focus on the Indian numbers. There are about 1.5 lakh deaths and over 10 lakhs have recovered. Okay, you can say, well, we are doing much better than many parts of the world, and we can have debates about this. And here is the status. These graphs I got from Waldemar last night. And you can see that the numbers really peaked at around September when people were getting tested. Probably not catching everybody who's infected, but whatever our mechanisms of testing, there was a peak then. It's come down, but it's still slightly over 10,000 cases a day across the nation. Deaths have come down as well. It's now a few hundreds of deaths a day. Okay, so overall, compared to where we were in September, things are improving. And is this a cause for celebration? Certainly, in some ways, it looks like a very positive turn of events. However, please keep in mind, there is something called people who recover from COVID. There are a large number of them who continue to have symptoms. And here is a study which came out not that long ago, last month, looking at patients who had recovered from COVID-19 6 months after they had been discharged. And this had been informally, in fact, there was a very beautiful article in The Atlantic by Ed Yong, I want to say as early as June, where he raised this issue, and there were other eminent people who worked in the infectious disease field who had raised this issue, and which is one of the reasons why I think one part of the reason why people in many countries have been very careful about managing the public health aspects. 76% of the people after they've recovered continue to show at least one symptom. 63% show fatigue or muscle weakness. 26% show sleep difficulties, 9% show palpitations. There's certainly some examples of heart abnormalities because this virus can infect multiple organ systems. Some number of people have anxiety and depression. One doesn't know if this is due to the act of the virus itself or other related issues, but certainly it's reported. And 50% have lung abnormalities. So you may be recovered, but you may still not have been restored to the health that you had before you had the disease 6 months hence. And this is in some sense in terms of looking at from a national perspective or a world perspective, many people, are they going to be back? Are they going to be able to work, provide care, deliver on their responsibilities the way they could before they fell ill? Is still out for debate, and these are somewhat disquieting numbers. And I think this also illustrates why we as communities and as people owe it to ourselves and to people around us to continue to follow best practices in terms of mitigating spread. Okay, what are the control strategies? Really, nothing has changed. In fact, many of these things, as I talked about isolation, for instance, was known with smallpox. We still do that in COVID-19. And many of these things came from looking at influenza pandemics, using masks, maintaining distance, hygiene, and if you're in an indoor situation, ventilation is crucial. So containment, test, trace, and isolate. So if you have tested positive, find the people whom you may have contact, isolate the people, make sure you break the chain, right? That is critical. Treatment, there are many things which are still companies like Merck, and there have been reports in Israel that there might be some other treatments which seem to have worked very well, which need to be studied greater. But really, dexamethasone is the star of the show given at the right time. And there are a few other things, for instance, IL-6 receptor antagonistics. Many clinical trials seem to say different things, so it's not clear exactly what the outcome is, but these are some of the treatments which are out there. But I think managing this disease is something that the world and all the doctors have learned to do better, and that has, of course, helped in the death rates and making sure that people aren't losing their lives. And all of these strategies, vaccines are what I call the probably something that we have all pinned our hopes on and is going to be crucial in the coming months. So what happens when you get an infection, any infection? This first response is what is called the innate immune response. It is for any infection that comes to you, it'll start right away. Your virus, you'll be very low, but if your innate immune response cannot control that load of virus, your virus is going to replicate, increase in number, and as it increases in number, slowly it will also trigger the adaptive immune response, which will rise. And it is these early events, which in COVID-19 of being pre-symptomatic or asymptomatic, which is also very dangerous because this virus then at that time has an opportunity to spread to others before you start showing symptoms and recognize that you have the disease. And by the way, this is again nothing new, right? I mean, people who, if you look at the older people in this community who may not have gotten vaccinated for some of the infectious disease, that, all these pustules and things that you got on your hand for measles, they have to fall off, because if you start going out, those will fall off and infect someone else. So that there was a period of infection that you could infect people at least afterwards when you're still not, all your sores are not dry. But clearly, it is very well known in the infectious disease field that many diseases have a short period of pre-symptomatic or asymptomatic phase, during which time you can infect other people. So it's by no means completely new. It's been known for polio, it's been known for other diseases. Okay, so what is this innate immune response? And it gets talked about, especially in India, when in the context of deaths and are we special people, I can address that in questions to the best of my ability. But this is a screenshot from Akiko Iwasaki's incredibly beautiful YouTube video explaining the innate and adaptive immune response. It's a very short video. I recommend it to everybody. What essentially the virus comes in, it's taken up, and it sort of starts a cytokine-based response, which can attract signals to which allow a cell which is infected to die, recruit immune cells to fight the disease, signaling neighboring cells to put up barriers. If too much of this interferon comes, then it's very difficult, and that is the beginning of what could well be a cytokine storm, although it can more typically occurs a little later. Often viruses have ways to attenuate this innate response and turn down the production of these cytokines and these other interferon-like molecules to escape from it, because obviously it's a race between prey and predator. The prey here is the virus, and the predator here is the virus, and the prey is us. And you sort of want to do this so that you can make more of yourself and infect more cells and move on from there.
Question, Sandhya?
Yes.
What are cytokines? We keep hearing about cytokines and cytokine storms.
So cytokines are small secreted molecules that come from cells, typically immune cells, but other cells can also secrete them, which are immunomodulators. For instance, here the interferon is a cytokine. It signals to many other types. So there are many types of white blood cells, right? There are B cells, there are T cells, or if anybody goes and gets a blood test, they'll get this neutrophil, eosinophil, all of those break down, right? They're all white blood cells, and they all do slightly different things in the immune system. They're all attracted to the site where the infection has, or maximum amount of infection has. But these cytokines also act on, sort of promote release of more cytokines, which can cause inflammation. And that inflammation is often the source of what is very, very difficult for the body to manage if it is uncontrolled, leading to bad outcomes. Okay, because your lungs fill up with fluid and other kinds of things, and then you don't breathe very well. I hope I answered the question. Okay, so let's look at what happens with a coronavirus infection. A virus comes in, it has this protein which I'm sure almost everybody has heard of, called the spike protein. It studs the surface. There are enzymes which sort of cut it, which are present on the outside of the cell, of the host cell, of the prey cell, and it allows this spike protein to present itself well. It binds to a receptor called ACE2 receptor, which is present in many, many tissues, including your heart, your blood vessels, your lungs. And this is what, in fact, has led to the SARS-CoV-2 virus being able to infect a lot of different tissues. It goes in, this virus gets translated, makes new viral particles, and gets released. So it makes more of itself, gets released into the bloodstream, infects neighboring cell, circulates and goes somewhere else in the bloodstream and infects some cell which is very far away. Okay, this is all pretty standard. What next? At some point, it will come close to something which is called an antigen presenting cell, which is take it up. All the proteins that it produces, it will chop it into little bits, present it on its surface. This is recognized, sends out alarm signals also sometimes. This is recognized by other kinds of cells called T helper cells, which in turn with B and other cells will make antibodies, which you have all heard of, the seropositivity case, or the T cells, which do a completely different way of killing, which is they destroy the cells which are infected with the virus. Okay, and I'll come back to this later when we discuss actual vaccine examples. This is the natural course of how your body would react against SARS-CoV-2, this aspect, or any other pathogen which will infect a cell. Okay, that's no different. There's nothing special about this virus in that regard about how your immune system responds, which has been really, really useful. There are no ugly surprises. We've pretty much done what you might expect it to do. Okay, so again, capturing it, you have an innate immune response, you have induction of immune response, which comes through B cells, which make antibodies, as well as T cells, which can directly kill cells which are infected with the virus. I won't go into a lot of the details because I think, in a sense, that's much more information that some of you may find a bit overwhelming, and we have far to go in the talk. Okay, so I showed you earlier the interplay between innate immunity, which is just what is actually innate immunity is present in fruit flies, which come to your banana in your house, or in C. elegans, which I work on. This is a very old immune response. The adaptive immunity is a more recently evolved immune response, which tunes itself to a particular kind of antigen. So you have your infection, it sort of peaks. You have specific T cells and specific B cells which come later. They all reduce. In fact, you must have heard in the news that seropositivity is going down. Does that mean you are going to be susceptible for reinfection? Your antibodies go down, but your memory cells remain. And your memory cells can come in two flavors, the antibody-producing flavors, as well as the T cell flavors. In fact, SARS, which 17 or was it 18 years ago, burst upon the world and caused a lot of deaths because, something to the tune of, I think something like 20% of the people who got the infection died, unlike COVID-19, where the case fatality rate is very low and the infection fatality rate is less than 1%. So it's very low. The SARS itself was not like that. The T cells against that remain 17 years later. And this is very hopeful information, even for this particular pandemic, because those two viruses are very, very similar. Okay, this was natural infection. What happens if you do a vaccination? Is it going to be completely different? Are we triggering something completely different? I know this looks kind of small. So you have a vaccine. A vaccine is some way to train our immune system to fight off a particular bug without giving us a disease. And this is very different from variolation or inoculation, where you can get the disease, okay? So vaccination is really to protect you, to train your immune system. So vaccine contains things like preservatives, emulsifiers, and stabilizers. It often contains an immune adjuvant, which can improve how your immune reaction is. It comes into, again, you have this particular antigen which you inject. It's taken up by your antigen presenting cell. And once you come to your antigen presenting cell, your pathway is the same. You have both your T and B cells being activated, right? And essentially, all your later parts remain the same, and you do not get the disease. Okay, let's look into why is it that we do not get the disease. We do not get the disease, and some of the oldest ways in which we worked. I mean, the smallpox example is actually a poor example in this, because smallpox was an example where you could train your immune system against a similar virus, which then cross-reacted and killed the smallpox virus, right? So initially, that's the way it was done. You can take a live, weakened, or inactivated virus. And in fact, often the cholera, initial cholera vaccines which were developed, initial typhoid vaccines which were developed, all followed this practice that you isolated the agent and you inactivate it in some way. You can kill the whole organism. You can use part of the toxoid. In fact, the adjuvant idea that you couple it with something else came in part from studies with diphtheria, because what you would do with the diphtheria toxoid is you would try to vaccinate someone with the toxoid, with a weakened toxoid or a partially killed toxoid, and the person didn't get long-lasting immunity because you were not triggering your immune reaction as much. So you combined it with an adjuvant and you got better results and better protection. You can have subunit vaccines where obviously a virus or a bacteria has large number of proteins on its surface, and you pick some parts of it to expose your immune system to it, which you think are the key to fighting off the disease. So in our case, in this particular epidemic, it is the spike protein which binds to the ACE2 receptor, which was the choice, because you know that that's very important for getting into cells, human cells. You can make virus-like particles, and you see some of the technologies are very low, live attenuated from 1798, typhoid from 1896, and from 1986, viral-like particles, which is much, much newer technology. And I will just say, if you look at the nucleic acid vaccines, the very first time an mRNA vaccine has been used in a public vaccination drive is starting last year with Moderna and Pfizer BioNTech vaccine, which are RNA vaccines. So this technology is constantly evolving. The viral vectored vaccine, which is the Covishield or the Oxford AstraZeneca vaccine, is also relatively new technology, first developed for Ebola and also used for Rift Valley fever, which is for animal diseases. So you know these platforms. So your new platforms are constantly being developed to improve your immunogenicity, improve safety, and to make sure that you have good outcomes. I want to touch upon this very, very important aspect. Why is vaccine safety important? Vaccines, we must realize, is given to healthy people. You don't want healthy people falling sick with some product that you're giving them. It needs to have high efficacy. If it protects only 20% of the people who get vaccination, it may not be as good as if you can get 90% protection, right? And by the way, efficacy in a clinical trial is very different from effectiveness on the ground. And I'll briefly mention it later again. It is often given to a wide range of people of varied health and ages. And I think that is particularly noticeable now with the vaccines against COVID-19. Less commonly, we have recognized it because most of our idea of vaccines is that we give it to children. Typically, multiple doses are given. And so again, safety is important. Maybe the first dose you did well, but maybe you didn't tolerate the second dose as well. So those concerns become very, very important. And last but not the least, This is a continuing, the last point is a continuing importance, the quality of production, the distribution, storage. Think about the Moderna and Pfizer vaccines. Pfizer needs to be kept at minus 80 degrees centigrade. Moderna needs to be kept at minus 20 degrees centigrade. Even our typical childhood vaccines, any parent who goes and gets their child vaccinated worries that, it's supposed to be kept at 4 degrees. I hope they didn't lose their electricity and have their inverter fail, right? These are all concerns that we have of storage, ease of administration. These are all important. And every batch of vaccine that you make has to be safe and efficacious. This is non-trivial. Non-trivial also from the point of view of public safety, but it is also non-trivial because you just need a few missteps and then you end up having vaccine hesitancy. Therefore, it's essential that vaccines must work as advertised.
So at
yes.
this point, can I just take two questions which have come in the middle?
Yes.
So Harsh wants to know why, you explained the different types of vaccines, but why are most vaccines in liquid form to be injected? Why not pills to be swallowed?
Oh, okay. Because your digestive system will chew it up, right? You have a lot of enzymes present in your stomach, which will just, oh, I'm so sorry. I wanted other people to hear it. But your digestive system will chew it up. So if you inject a protein adjuvant vaccine, it has bits, say, let's say you have subunits like this, soluble proteins, which are subunits. You put it in your stomach, just as you digest your food, you will digest this protein also before you trigger any immune response, which is one of the reasons why, but please note, there are some which come, oral polio vaccines come in that way. So it really depends on the platform and how you evolve it. But those are issues that need to be dealt with. The oral polio vaccine, if you remember, is given in your mouth and it still gets through and you can still trigger a response. But that's not typically true. The typical thing is an injection.
Okay, and while we are at it, let's take a question from YouTube also, since we've anyway interrupted. I mean, this is, of course, from the very beginning. Why does the pathogen sort of cause a lethal effect on humans? Because from the pathogen's point of view, if the host survived for a long time, it would probably be able to replicate a lot. If the host dies, the pathogen also has to die along with it. So, why do these viral infections sort of make us sick? Wouldn't it be better if we were not sick and the virus would continue to spread?
It's a very good question. Any pathogen which attacks you ideally wants to live and wants you to live so it can spread to others. But independently, that is why I briefly mentioned the SARS-CoV-2 actually has a very low infection fatality rate. Okay, it is less than 1%. It's like 0.08% or 0.09%. So it is very low. It is just that this particular virus is very infective. It spreads very easily. In fact, it doesn't compare to the measles virus. There's something which you might have heard of, which is the R0. The measles virus, one person who's infected can infect 18 other people. The SARS-CoV-2 can infect, between 2 to 3 people. And in general, you will find in certain cases, the pathogen does evolve. And as you can see, you must have heard about the variants of concerns which were detected in United Kingdom and South Africa, as well as in Brazil, that viruses do evolve. They change. They can change in how they infect people. They can change in what are the disease outcomes. And I agree that usually if the infection will be self-limiting if all the hosts who get infected pass away. So that process of evolution where you have a race between how the virus is when it emerges and how it continues to progress is going to happen only if there is a sufficient reservoir, because the virus, as it replicates, any virus, any bug, as it replicates, will accumulate changes within it, which will allow it to have this arms race take place. And this is not just with a virus infecting some human being. It could be a virus infecting cattle. It could be two different species competing for the same resources. This is evolution in action. In fact, there's this very nice paper which had come out in eLife, I think early January, where they were discussing issues of vaccine escape and things like that. And they were like super excited. Oh my God, we are seeing COVID-19 undergoing evolution in action. This was in a dish, okay? But nonetheless. And so this is part of the natural process that happens in the world. And I sort of talked about the issues at hand rather than saying that, this is what will happen, because all change is chance. What remains is what is acceptable. So unless there is a selection pressure in some way, all changes will be tolerated, would be the general way of thinking about it.
All right, thanks. Great.
Okay, so let's look at what goes into deciding safety and efficacy, because I think that's very important to understand. Okay, so how is a vaccine developed? You first have some preclinical trials. You figure out whether what you have made can work in cells in culture. Does it work in maybe small animal models? If you have a reasonable animal model, which is a mimic of the disease. And then you have phase 1 trials when you come to the clinical aspects, where you have very few volunteers, maybe up to 100 volunteers. And the only thing you're seeing is it safe? And are they suddenly falling sick? Do they have really adverse reactions? And is there anything, is it related to the size of the dose? How much dose should I give? And let's come back to this. We had this dosing issue with the Oxford AstraZeneca, where when they had a half first dose and a full second dose, they got much better efficacy than when they had 2 full doses, which, by the way, is what is being offered in India, as far as I understand. So there are many issues like this that need to be worked out, and some of those are addressed in phase 1. In phase 2, where you have several more volunteers, again, what you're trying to see is, do you have an immune reaction? Are you still safe? Because what is 100 volunteers? If you have something occurring at 1%, some site of side effect at 1%, which is very high in vaccine land, okay, the adverse effects that the Pfizer vaccine, the allergic Pfizer, anaphylactic shock Pfizer vaccine, was initially thought to be 12 events in 1 million people. Now with better data, we know 6 events in 1 million people. This is nowhere near 1%. But if you're testing only 50%, it's not a given you will catch the 1%, right? Safe, it is that high. So you need the phase 2 trial with several hundred volunteers, or the phase 3 trial is when you begin to see whether it actually works. Okay, the phase 2 trial will see whether you have an immune response, and if there are against safety trials, additional safety trials that you will watch. But it's here that you're really testing in phase 3 whether the vaccine is effective. And that efficacy is a very important step, because what you do that is in a controlled environment. There are 2 arms. There are people who get the vaccine and people who don't get the vaccine. The disease is going around in the community, and then you say, okay, what fraction of the people who got the vaccine did not get infected or did not get the disease? And then you look at the control arm who've got the placebo, and then you ask what their outcome was. Did more of them get it? And that's very important. Here's where the issues also came up as to whether we should do human challenge trial, because so many people are getting infected. Maybe we can get more direct data instead of letting community spread decide what is happening. Okay, so there were other ethical issues associated with some scientists being very for trying human challenge trials, and others saying we don't have good treatments, we have this phenomenon of long COVID, we need to be careful and we need not do it. So you can see how complex it is. It is not just science. Vaccines are closely interrelated with how societies work, how politics work, and the values of a given society as well. Okay, are you done with phase 3? We are all, I think, at this point of time, knows things about phase 3, and we said, okay, we are at phase 3, we've done well, we have good efficacy, some 60%, some 90%, let's put it out in people. Is that where it ends? The answer is no. You have phase 4, which is post-licensing, because now you will be vaccinating hundreds of thousands of people, especially in an epidemic like this. And you continue to have reporting mechanisms. You continue to see if it works well. And if you have new variants are coming, is this vaccine still working? These are all very important questions that people need to keep in mind. So vaccines and making effective vaccines is a moving goalpost. Some of you may already know that for influenza vaccines, you make a new one every year because you have different circulating strains. So you have a platform that works very well, but every year you have to update it, right? So there are many, many issues associated with this that one needs to think about, and safety and efficacy is at the center of deciding whether a vaccine moves forward or not into the clinic. Okay, here I thought some of you might have seen it. I myself did not know much about Dr. Haffkin, and I thought it's a kind of nice story. It illustrates a couple of points. So Haffkin was a Ukrainian or Russian at that time, who, because he was a Jew, he could not practice where he had got his degree. He came to France and he joined the Pasteur Institute, and cholera was a huge problem in the world. A lot of people were losing their lives. He developed an attenuated vaccine against cholera, which he tested on himself and a few of his Russian fellow mates and volunteers. He felt sure it worked, but there was resistance to trying it in the field, because essentially what he had done was phase 1. Little bit of bias maybe in his choice of people, but what he had done was a phase 1. But he needed larger numbers. So at that time, it was suggested that he go to India, because India had a lot of cholera cases, and to try out his trials over there and see if it worked. So he came to India. He conducted large trials in Kolkata. He sat himself and inoculated people. And this is from the BBC article where he's inoculating people. He inoculated himself in front of people to sort of, again, convince them that you should take this vaccine. He was able to do these wonderful control tests. In a family living in the same place, he would inoculate some, he would not inoculate others. And he started getting data. He was looking at clinical trials, which you're doing in the 1800s, where you see is that people can be in the same family. The ones who got inoculated did not get the disease, and the ones who did not get inoculated can get disease and can die. So he started finding out that the cholera vaccine actually worked. What he saw was it really reduced infections, but not mortality in those who got infected. So despite having the vaccine, you can sometimes get infected because it's not 100% protection, right? And if you got the infection, it did not reduce mortality, which is very different from what we are seeing with the current COVID-19 vaccines. These are older technologies, obviously. So he wanted to go back and rejig the vaccine so that he could have a better outcome. Okay, so then he went on to develop a vaccine against plague in Bombay. He did trials in Baikola prison, which is in Bombay, and we are now in Bombay. And in fact, his plague vaccine, there were some missteps in his career, which we'll not talk about, which is one of the reasons he doesn't get talked about as much. And they were not his fault at all. There was somebody who was delivering the vaccine had contaminated the vial in some way, and there were some deaths. But unfortunately, that sort of blew back to Haffkin, and it took many years for him to clear his name. But overall, 26 million doses, with 50 to 80% reduction in mortality, which is a huge public health win against a really scary disease at that time, which was plague. I also wanted to mention that vaccines do fail. And this kind of inactivated technology, which was popular at that time, 1962, inactivated measles virus just failed. It did not work. It did not provide any protection. So this is why safety and efficacy and design of clinical trials has always been the mainstay of taking vaccines from the laboratory to the clinic. Okay, that's something to keep in mind and appreciate, and how difficult it is. So let's step back again. What's the ideal vaccine against SARS-CoV-2? We want it safe for all age groups, efficacy in all age groups, safe for all groups. So if you are a cancer patient, it should work for you. If you're pregnant, it should work for you. By the way, trials have not been done for these groups of people, so we don't know anything about the current vaccines and if they will work. It would be ideal if vaccinated people do not shed the virus if they get infected. So they have a there's such good immunity that they don't replicate much and it doesn't go out. It would be great if it's single dose. It's very difficult to get people repeatedly. It would be nice if there was no requirement for cold change or syringes. In fact, that was the question which came. An oral vaccine would be fantastic, like oral polio was, and can be manufactured easily, right? These are all key things for an ideal SARS-CoV-2 vaccine. And in fact, for any vaccine, right? But in this case, we really have so many people in the world who need it. So all of these become especially important. How do you decide if a vaccine is safe? Immune reactions against the vaccine is important. That happen for the respiratory syncytial virus, where upon getting vaccinated, and the way you generated certain kinds of surface proteins which allowed, or it happens with dengue with different strains as well, dengue vaccines, when you get the infection, you actually, the viral particle enters cells more. So you get a worse disease. Does not seem to be happening with the SARS-CoV-2 vaccine so far. It's a very standard vaccine, a standard bug, which is very good. Sometimes you have unintended consequences. There was an Australia vaccine, which was a subunit vaccine, which has a small bit over it, which was part of the HIV, it was part of the vector. Now these people developed an immune reaction to whatever was provided, and it's a spike, but also against this little bit which comes from HIV. It had to be stopped, because if these people later on got tested for HIV, they would test positive even though they don't have HIV. So that trial was stopped. Side effects, very important, and we continue to catalog them. Some things are coming out in the news even now, so you might be paying attention. Adverse effects, are they short term? Are they late onset? Is it rare? I already described what happened with the Pfizer BioNTech mRNA vaccine, which is a new platform. So when you have new platforms, you have newer adjuvants, newer kinds of materials that go into stabilizing it. You don't know if it's just the mRNA, it could be the other components which are in the vaccines, which are critical and important. So I already told you that it's 6 anaphylactic shock events in 1 million people, but we need those numbers to know really what we are looking at. We also need background information on rates of adverse effect. If you're saying that some people are getting, their arm is swelling up twice the size, is it normally swelling up twice the size for other people? Without even getting the vaccine, do you have something out there which is giving this kind of reaction? So you need the background rate of adverse reaction in the population for you to recognize whether this effect that you're getting is because of the vaccine. It could be something completely unrelated. You need different age groups and how does it behave in immunocompromised people. Again, important information to know when you're doing vaccine efficacies. And of course, we have new technologies, vehicles for delivery, newer adjuvants, age-dependent efforts, all going to deciding if something is safe, which is why vaccines often take a really long time to make, because you have to get all of this information. How do you decide if it works? And you decide if it works if you get an immune response in your preclinical and your phase 1 and phase 2 clinical trials. And in fact, 2 viruses have been abandoned because they did not generate enough of an immune response, which is the Sanofi GSK subunit vaccine and more recently the Merck viral vector vaccine. Okay, dose range, that is sometimes decided in phase 2. I already mentioned what happens with AstraZeneca. Full dose, only 62% protection. Half dose, full dose, 90% protection. Why is that? We have some guesses, but no real answers. But again, that's information that comes from clinical trials. Actually, the full dose is what has gone through the entire clinical study, and therefore, 62% protection is what you can expect with this vaccine. Okay, is the immune response protective? What we already know from looking at, and I'm sorry to have all these text-heavy slides, but I didn't quite know how to do it with pictures. Is the immune response protective? And the answer is, what is protective? We already know from looking at patients there's a wide variety of immune reactions possible. Some people produce a strong immune response. Some people do not. It seems to depend on viral dose that you get and how much your virus replicates within you. There is that variability inherent in real infections. What is the correlate? How can you say that it is being protective? This is an extremely difficult scientific problem. People now think that the correlate of protection is if you can prevent the virus from entering into the cell. But how do you test that? It's not easy. It's not just a blood test that you do, because you actually have to do a test where you say that it cannot enter into a cell. And is that something which is a good correlate? Does that also hold true in human studies? They're tantalizing indications that might hold true, but to do it routinely in the field, very different from doing it in a specialized lab, right? So those are also very important. How much does it protect? I already said the design of clinical trials and how that's done. How well does it protect in regular use? Effectiveness. Because now when you're going out, you're going to see the full diversity of people with all their comorbidities, with all their age ranges. And you don't know if you would have done efficacy in a small group of people, like here from 18 to 65 years of age. We have no idea if you're 18 to 65 but have comorbidities, or you have some other issues with your health. Does it still work as well? So effectiveness on the ground is going to be very different. And you can also assess that by saying, have you got herd immunity? So I will finish fairly soon. So studies are decided that are you going to reach herd immunity? Studies to decide when you give a booster and how long does the protection last. How did it get here so quickly? Usually it takes 10 years to get a vaccine in, sometimes 20 years. How did we get here? Several technologies were well established. MERS and SARS, we had similar viruses. The vaccine target, which was the spike protein, was known. Some preclinical and clinical efforts ran concurrently. There was money pumped in from multiple sources, including companies. Companies and regulatory agencies worked with urgencies. Production capacities were ramped up before efficacy was known. Serum Institute made hundreds of doses before the phase 3 trials were out, and whether it worked or not, right? So people bet. In fact, Dr. Fauci said in several interviews that, the US has taken that on because we have built a lot of manufacturing and stocked up things simply because we want to be ready if something works to roll it out. People have been willing to volunteer for back-to-back trials. And for some vaccine candidates, all steps were followed and data and interim data is available for independent review. And that's a very important criteria for deciding whether a vaccine is good or not. So, as I said, these are the different platforms. What is the situation in India? The green are what are in clinical trials. The red are in preclinical trials. And as you see, either in manufacturing or in developing our own, we have several things which are up and running. So Zydus Cadila has a DNA-based vaccine. Serum Institute, you already know, has a viral vector. Reddy has tried up with Gamalaya to make the Sputnik 5 in terms of manufacturing. And many of these which we are getting from other countries, like even the Covishield, you have to do a bridging study to make sure it still works appropriately in our community, right? So this is the full spectrum. India supplies 60% of the vaccines in the world, and so we are in clover in this case, and we have an opportunity to protect our populations, but also to help other people in the world. Okay, so let's briefly look at Covishield and Covaxin. Covishield is an adenovirus platform. Your little spike protein is present inside this adenovirus. which enters the cell. This DNA is injected, it makes mRNA, makes all these proteins, comes up on the surface, and these proteins, the cell can die, the proteins will be released, then these digested proteins will be presented on the antigen presenting cell, attract the adaptive immune cell, and then start immune chain and protection reaction. Okay, what happens with Covaxin? You have essentially the coronavirus. You inactivate it using chemicals so that it by itself cannot cause disease. And you have to, of course, test that. But it also means you have to grow a large amount of live coronavirus. So you need very safe places to do that. You grow this, you inject it. This entire particle is taken up by these antigen-presenting cells. It presents these spike proteins on the surface, and then you have essentially a common pathway where you make antibodies from B cells. You stop the virus by having these antibodies coat the virus and prevent its entry into human cells. And also, if you have infected cells, your long-lived T cells will come and kill the cells which are infected. So there are two parts. There's a cell-based immunity, and there is the antibody-based immunity. I have now a portion of the talk which is just easy listening, and it's some issues that we need to think about with respect to vaccine. There are many major vaccine initiatives. I don't want to read the slide. I just want you to look at this red box. COVAX is very important. We are in India getting 97 million doses from COVAX, which is basically a grouping of WHO, CEPI, Gavi, to come together to bring countries together to buy into COVAX so that you can negotiate with companies for good prices and have equitable distribution of vaccines. So you generate money, you sort of do this public health effort. And the idea is a very simple one. Nobody wins until everybody wins. And this holds true for an infectious disease which is in pandemic mode, which is present all over the world. Because you might protect your population more or less, but there may still be small gaps here and there. And if you have someone who comes in with a new variant from another country who has not been vaccinated, all of that could be for naught, right? So in terms of public health measures, the COVAX facility is a very important initiative. This is ongoing across the world. Vaccines that are approved, you can see, and I'm going to highlight here Israel because I want to show you that the coronavirus vaccines we think work. These are at least in some country, these are the mRNA-based Pfizer BioNTech vaccines. And here are the two which are available to us. The AstraZeneca, in India, it's known as Covishield, which is also approved in a lot of countries, and Covaxin, which is approved only in India. So you see that there is a race with the BioNTech being the one which is approved the most. I just wanted to again highlight the standard dose gives you 62% protection, and the half full gives you 90% efficacy. Okay, some vaccines are in phase 3, and these are late runners, right? So here you have Johnson & Johnson and Novavax. Johnson & Johnson has been shown to protect. Here, this is the first vaccine which can protect as a single dose. So somebody doesn't need to come in again to get their second dose at the right time, has 72% efficacy. Novavax is 89%. Now, everybody knows about the variants. No problem with the UK variants, but efficacy drops against the variant identified in South Africa. So again, we are already seeing that some of the vaccines we have developed may not work as well against the other variants. So you can get infected, but you can get infected. One doesn't know, and one hopes that you don't progress to disease or severe disease. So there's a difference over there. Many vaccines still in development, 16 are in phase 3 trials. Vaccines come, as I said, some are one dose, many of them are 2 or 3, 2 doses, which are 21 days apart, or 2 doses, which are 28 days apart, which is considered a prime boost of the same kind. And majority of them are subcutaneous, intradermal, or intramuscular injectables. There are only 2 which are of the oral variety. Okay, so this is the landscape as it exists now. Every Friday, WHO updates this file, and it's available for public access, and you can download and look at it. Okay, what has WHO approved? Because that's what is going to be bought into the COVAX facility. Okay, the only vaccine to date that WHO has approved, I don't know why my computer is becoming crazy here, yes, is the Pfizer. Everything, it is anticipated decision, it has already been approved in the end of December. Serum Institute, which is probably of interest, will get approved mid-February. And you can see, for instance, the Sputnik 5 vaccine, they have sought additional information. There are several consultations gathered. Rolling data is expected from trials, which they will look at, the whole committee from across the world on middle February, and then they can say when it will get approved, right? So everything is coming in, but it's not all there. In fact, last year in June or so, they'll say nothing until mid-2021, and that's what we are seeing. Maybe a little bit, 1 or 2 are early in the game, but by March, the world over picture will be much clearer. Why do you even bother with so many vaccines? Need is high. We need single dose administration, maybe better protection, maybe work against the variants, maybe you can scale up production much better. These are all reasons why many vaccines are still in the pipeline and companies are still in it. Some, like Merck, have said, no, we're not going to do vaccines anymore. The one we were trying to develop did not have sufficient immunogenicity. We're just going to focus on therapeutics. I just want you to consider what is happening. These are high-income countries, and they have pre-ordered vaccines, taking bets, agreed, which will cover their population 4 times maybe what is necessary. That's Canada, that's US, which is almost 200 times of their population they can cover. And where does India lie in this? India lies at around 60 or 70%. So vaccine nationalism, the power of being a rich country is all coming into play, and it's something that we need to be aware of as people who are going to be consumers of the vaccine. Yes.
I think you mentioned that India is one of the largest suppliers, the producers of the vaccine.
Yes.
And still we are not covering the whole of the population.
Can you repeat the question?
So the question is that we are India, as I said, synthesizes 60% of the doses of the world, and she's saying that why is it that we have not pre-ordered vaccines for 100% of the population? So for that, to that level of pre-order, you're going to need 2 things, I think. I mean, I don't know all the details, but 2 things I think you're going to need to have that all being developed in India. And as you already know, Serum Institute has promised us 100 million doses at 200 rupees a dose to the government, but they need that serious money. And we don't have all of these strains, all of these various vaccines which other countries have bet on. The ones which are out there are the mRNA vaccines, which we don't have yet the means to generate in scale, manufacture in scale in India. We manufacture the adenovirus vaccines, the inactivated vaccine, the DNA vaccine, Zydus Cadila, is one of the first of its kind, which is, if it comes out and pans out, would be very useful. But we don't, the vaccines which are out there, which are in the forefront, no. And you know what happened recently with the EU, right? They told AstraZeneca that, what's promised to us has to come to us. So there is some, give and take happening here. It's complex problem. And I think that's something that we as consumers of vaccines need to regulate. Israel is way ahead. Close is United Arab Emirates, but United Kingdom is very good. India is less than 0.5%, okay? So what is the data from Israel, which is the only country where you have vaccination taking place, where 54, this is not the number of people who have been vaccinated. This is the number of doses that have been given. So you need 2 doses for a vaccine. So you see in Israel, what you saw was people after this level, early in February, over 60 years of age, 90% were vaccinated with 1 dose, 41% there was drop in infection, 31% drop in hospitalization. Not as much in less than 60 years because not as much vaccination either. But these are the first indications that vaccination can have countrywide effect on spread of infection, which is very, very good news. We didn't know this, and we can't take an assumption that that happens. We need to know that it so works, and here is our first data.
I mean, despite India not being vaccinated so much, there has been a huge drop in infection, right? So how much of that could you attribute to the vaccination?
I don't think that it's too soon in India for the vaccination to have had an effect.
No, I'm not talking about that. I'm saying that without vaccination,
yes,
India
we already, will vaccination have the same dramatic effect that we see in Israel for infections? Is that the question that you're asking?
Was that how much of that could be attributed to vaccination in Israel? When you see that without vaccination, India has seen a huge drop.
So this is comparing data between pre-vaccinated, which is December, to what is happening end of January. So this is literally 1 month apart. So it is a reasonable thing to say that, so you have two kinds of controls, right? You have the above 60 years showing this big dip, which is the most vulnerable, and below 60 years who have been vaccinated less, showing less of an effect. So given that these data points are about month or two apart, it is very likely that the effect is on vaccine, is due to vaccines and not due to just natural drop in infection. Okay, so we are in the last two slides. Vaccines are not a magic bullet. COVID-19 vaccines so far protect against disease, but not against infection. So you can still get infected. And while you might not get a severe disease, someone you come in contact with who has not had the vaccine could very well acquire it from you, right? Which is one of the goals, that you don't want people to shed the virus if for an ideal vaccine, but that is still not completely clear. But what is clear is these kinds of vaccine will protect against disease, but not infection. So mitigation of masks, distance, hygiene, and indoor ventilation, when you're indoors doing ventilation, are going to be critical. Containment, test, trace, vaccinate, isolate, especially with newer variants in the horizon, which appear to infect and spread much further. This is going to continue to be important. Treatment, the ones which I said earlier still apply, and there are many ongoing efforts by many people. And vaccines, we are hoping that you will have herd immunity through vaccination. The Israel data, which is preliminary and suggestive, looks as if we are going the right direction. We'll have to think about vaccinating beyond priority groups. What are we going to do about vaccinating recovered people? All issues to consider. But vaccines are not a magic bullet. I'll end with this. You might have heard, and I certainly have heard from various people, that they don't plan to take the vaccine. And they just say that, I don't plan to take the vaccine. Why? COVID-19 is not a dangerous disease, but it may not, you may think you may be spared, but we still don't understand why is it that we know at a population level, some people above a certain age are very, very sensitive, and a larger portion of them pass away. We know comorbidity, people with comorbidities have a higher fatality rate, but we can't predict that an individual in that group will recover or pass away. We've had young people pass away. We say, oh, there's very low likelihood of that. It's not zero, right? So it's not a dangerous disease, but at the population level, we are not able to predict what an individual, we can only predict at a population level, we can't predict individual outcomes. Some people are concerned about side effects and safety, which is why the clinical data is very important. So far, the two vaccines offered in India seem to be safe. Those tests and ongoing vaccination effects suggest that they are relatively mild effects. Vaccine can cause the disease. Certainly for Covaxin, it's not even possible for the adenovirus based vaccine that it will cause the disease, because you're not even using the whole virus. And there are no vaccine-related immune responses that have been reported so far. And the Covaxin is inactivated, so it should not and will not cause the disease. Some people are worried, even though the nucleic acid vaccines are not offered in India, that it will change my DNA. No, it won't. It's a small bit. In fact, our body is infected with viruses, has endogenous viruses that we carry, which come to us cold. We get cold viruses. Our cells are not being changed by that as far as we know. So it's something which came up when we were speaking to volunteers through the Bangalore Science Gallery. We were training on ground people. Vaccines are for children, adults don't take them. Not true. In this case, adults do need to take them. In fact, it appears that certain age group children may have better outcomes than even adults. So it's a good thing to take. Vaccines are needed for all ages and stages of life. And some of them are worried about the cost, but we have close to 200 million doses, which is going to be publicly available, which from COVAX and Serum Institute. And I'm sure as other vaccines come online, I am sure that those companies in turn will offer the Indian government doses. So hopefully it will be available for majority of our people at an affordable rate, if not free. And thank you for your attention.
Okay, thank you, Sandhya. And there are tons of questions, as you might have expected. So we have people on the Zoom call with questions. We have people on Facebook and people on YouTube. Given that we haven't taken a question from Facebook, can I start with a question from Facebook?
Yes, please. You hear me well, Arnab?
I can hear you perfectly well. I hope the audience can hear you well as well.
Remove my earphones so the people in Prithvi Theatre who have been looking at, so maybe you got my profile, can also hear the question.
Okay, but so you will repeat the question?
No. I've just removed my earphones so they can hear from my laptop.
Okay, if it's loud enough. If not, you'll have to repeat it.
Loud enough? Yeah, loud enough.
Okay, so there is one question which is common to Facebook and YouTube. This probably is a very simple one, so we'll start with this. What is a toxoid?
Toxoid is basically a toxin which sometimes, like with diphtheria, what happens is the bug releases a toxin, which is what causes all the problems for you. So a toxoid is essentially a toxin molecule, a small toxin molecule.
Okay. Now, I guess I will take the more politically difficult questions at the end. Let's do the science questions first.
Yes.
Okay. I think people are still not clear what is mRNA technology, how is it works, what is a virus vectored vaccine? Virus vectored vaccine. This whole mRNA, the more modern vaccines. Maybe it's worth revisiting that once.
Absolutely. Let's revisit that here. Okay. So I'm still shared, right?
Yeah.
Okay, excellent. So this is a viral vectored vaccine. So let's get the whole slide up. So this is an adenovirus vector, which causes cold, by the way. Okay. And in its DNA, you remove all the parts which are not necessary, except the part to make this viral vector. And in this DNA, you incorporate, in the adenovirus cold vector DNA, you incorporate a sequence, a DNA sequence, which encodes for the spike protein. And now you grow this viral vectored particle, which has this modified DNA within it. It can grow. You make a lot of this virus. Oh, I should do this. Sorry. This is better. You make a lot of this virus and laser pointer. You make a lot of this virus. You inject it in your dose. Then this entire viral particle comes in. This viral particle will inject its DNA into the nucleus of the human cell. This DNA will make RNA, and this RNA will come out into the cytoplasm, get translated to make a lot of spike protein. But in case of Covishield, it's, some of them are replicating, some of them are non-replicating. We need not go into it, but you will not make more of this particle. It's just the initial infection which will give you this mRNA for the spike protein, and then you make this large amounts of protein. What the mRNA vaccines do is you directly inject the mRNA with particular kinds of components with it which allow the mRNA to get injected or cross the plasma membrane and enter the cell. Now this mRNA is directly injected, which is why if you have variants, it can be easier to deal with the mRNA vaccine, which is why it was also faster, because you don't have to make a virus, or you don't have to grow like Covaxin, a large number of COVID-19 virus itself to inactivate it. You just make this little fragment of mRNA in large amounts. RNA has to be kept stable. It can degrade, but there are technologies to do that. And then you just inject it. This RNA enters the cell. So you directly at this point, you bypass all of this, and then you make a lot of spike protein, because what you're injecting is the RNA for the spike protein. This spike protein then gets onto the cell surface. Sometimes there are enzymes up there which will cleave it, which will go out. Sometimes it will get secreted through other processes, and sometimes when they present like that, other cells will come and kill it, or the cell itself will die, and these viral proteins will then be circulating. And then the next step of the immune cell, the antigen presenting cell, comes in. I hope I answered the question.
Yes, I think that was good. Let's move to another one, which says, okay, if I get a, let me just try and see. Vaibhavi is asking, if you take a vaccine for a specific virus, is it possible that this whole process of developing antibodies, etc., would make you more prone to getting some other infection, perhaps? If I understand that correct.
No, you do not, right? For a normal, healthy person, absolutely not, because what you're doing is your immune system is constantly getting educated. We are constantly getting some bug or some kind which is coming. You may not get symptoms, but you constantly are getting things from the air, from the water, from the surfaces you touch when you put it, the cell phone that you, God knows where you place and what you touch, things are coming. So your immune system is constantly being challenged. And that is part of what the immune system does. Just because you're reacting against one kind of infection or one component of the protein which is coming through a vaccination does not compromise the ability to act against other infections.
Okay. There are a whole bunch of questions sort of related to the theme of the comparison between different vaccines. So, for example, if you take some vaccine type which is being given in country A, okay, and you travel to country B, are you still safe? So this is one kind of question, if you want to repeat it. Also, I mean, why does every country want to test its own people with the vaccine and not rely on tests done elsewhere, is a similar question. And why have vaccines not been tested on children and pregnant women and groups like this? So I think these are sort of related. So I'll take them together.
So I'll sort of try to answer all three questions. I hope it's reasonably satisfactory.
I hope the audience in the theater could hear those questions.
No, they could hear the questions. Yeah, they can hear the questions. My laptop is very loud. The first is, currently every vaccine has slightly different efficacy, right? Reported efficacy. So the mRNA vaccines from Moderna and Pfizer are over 90%. And the one which is Covishield is reported as 70%. So obviously the number of people, the extent of protection between those two, the number of people who will get protected or, is going to be slightly different. Not number of people, it's how well you're protected against the disease is going to be slightly different with these two vaccines. It does not matter what vaccine you get, as long as you have the safety and efficacy data available, especially the efficacy data available, you will have protection. Once you have that vaccine and you travel to another country, you don't have to get a new vaccine. You've already been vaccinated. Now, if the strains change enough such that the protection really drops low. And in fact, I showed you that for the Novavax and Johnson & Johnson's, they have already done those trials. And although the numbers are low, it appears that something which is close to 70, 90% in one case drops to about 50% with the variants that are coming from South Africa. Almost no effect on UK. So this is going to happen. Today we are talking about variants coming, which have been reported from these countries, but there are other variants which are going to come simply because such a large number of people have been infected. So the virus is replicating in all of them. It's changing in all of them. They in turn are going to infect some number of people. So these changes will then propagate in the population if it provides an advantage to the virus, right? In this case, can it infect more people? That is part of the process. So the vaccines may need updation. We still do not know how long the vaccines protect. It might be the flu, like the flu vaccine, that you need a yearly vaccination. And if you do, then you'll have updated vaccines against then circulating strains to do that. What was the third question? Oh, why do you have bridging trials? No, mostly countries, not all countries need bridging trials. India seems to want bridging trials, which is just small to make sure that in our population, with our kind of immune education and our local issues, these vaccines still work. It's not unreasonable. And in this case, I think in some cases they are just doing very, very small trials to make sure that you're not having something unique happen. Remember, always with a vaccine, safety is a key issue, right? Followed by efficacy. So you want to make sure that that boundary or that line is always crossed, that you know for sure that it's safe. And that is one of the, I think, motivations behind doing these very small bridging studies. They're not going to give you a whole lot of data. And if you have some emergency use, then those kinds of bridging studies can also in certain cases be speeded up or said, we will await it, but let's set the distribution in place. So no, some of these vaccine trials which are being available, like the Covishield one, has not all been done in the UK or in the EU. They've been run in other countries and they have taken that data. So by no means you are silo saying that unless we test it in our population, we are not going to do it. But in our country, we have, of course, some concerns that should we be thinking about, our own population and making sure once more that we are safe. I think that's the primary role of a bridging study.
So you've already alluded to it, and there are three questions. There's Samhita, Surit, and other people asking, Swapnil, this whole thing about how long will the sort of immunity or whatever you call it last? Are you going to need booster doses every year? Why do you need booster doses for some vaccine? Why is it not lifetime immunity? And also there is a thing about, is it better, is an mRNA kind of vaccine better for long-term immunity than an attenuated virus vaccine?
We do not know. Answers for all of this is not known. I was just telling, I think I said this in the beginning and I'll repeat this again. We've had this disease for a teeny bit over a year and we've made tremendous progress. But to know how long a vaccine lasts, you must have the vaccine, and the person who's vaccinated has to be in the community around for us to know. We are now getting data, what happens in natural SARS-CoV-2 infection, and we say 8 months hence, antibodies seem to going down, T cells are going down, they're going down at different rates, but the memory cells seem to be still present. And I told you about SARS itself, which is the coronavirus version, CoV-1. And you had the T cells which could kill cells which are infected with the SARS virus or CoV virus 17 years later. So if you look at that data, you would say, that's good news. And is it going to apply to SARS-CoV-2 as well? Because they're very similar. If it does, we are in good shape. But how long will the vaccines last? Your guess is as good as mine. There is
Okay.
a question in Prithvi. Shall we take that?
Yes, please. Yes, please go ahead. Do repeat it for the online audience as well.
Okay, so this is a very, used to be a very popular question early on. He's asking, there's conspiracy theory saying that SARS-CoV-2 is a man-made virus. Could it be a man-made virus? The answer appears to be a resounding no. Okay, so it has similarity to circulating coronaviruses in bats. It seems to have picked up something from pangolins. And one doesn't know when it jumped to humans, but there is certainly precedent of coronaviruses jumping to humans. MERS is a great example. Every once in a while, MERS jumps from camels to humans. And it's self-limiting. It has a very high mortality rate. Not self-limiting, it has very high mortality rate. So people who get it, a large number of them die. But there is precedent in nature for that. And there seems to be absolutely nothing in its sequence that suggests that it was made in a laboratory. And I think we should take this on board seriously. I know there have been these conspiracy theories, very unfortunate. Shall I take one more from Prithvi? Okay, yes?
There are other conspiracy theories saying that vaccines cause autism. Is there any actual correlation of the same? And what is the origin of this conspiracy?
Okay, it's a question about, there are these theories, conspiracy theories that vaccines cause autism. And is there any substance to it, and what is the origin? The origin was a paper which was published in Lancet, which was then withdrawn because of flawed data presentation and analysis. And many subsequent studies have looked at these issues. There seems to be zero correlation between taking vaccines and autism. And I think this should be debunked as much as possible at every level. So far, the vaccine effect seems to be protective. Consider a life without that. How much time, how much loss of life, how much long-term health effects, how much the fact that people cannot work at their full potential or be available for their families and their communities. The loss in not taking a vaccine is very high. There are certainly side effects of what are minor side effects, which we would consider of fever, arm swelling, joint pain, maybe headaches. Those are common. Some have it, like DTP tends to give more of that than others. There have been severe effects reported, but they are very rare. And this is why the phase 4 post-regulatory approval is also very important to collect and report that data. But autism, no. Please debunk this anywhere anybody hears it. Yes.
Okay, my suggestion is, can we finish the online? There are a few more questions online.
Yes.
We will finish. So my suggestion is we will finish the few questions that are there online, and then we will stop the live stream, and you can continue the Q&A session there with the people. That will be a much more intimate thing without having to worry about live stream, etc.
Yes.
So with that, I'll get the more difficult questions now. The first one, obviously, which you expect probably is, what is the phase 3 status of Covaxin, and what is the WHO status on Covaxin, etc.?
Oh, I thought I already showed you where the WHO, I mean, that was part of the reason why I did show the WHO data. And yeah, here is WHO. They have only approved Pfizer BioNTech so far. And I leave this on. You can see it on their WHO site. It's a really useful thing, and all of the WHO documents are updated regularly. And for those of you who are interested, they do a weekly press conference, which is streamed on Periscope and extremely thoughtfully done, giving you the current information. So it's anybody can access those. Okay. Covaxin, we don't yet have phase 3 data. I assume it will come soon. It certainly has been shown to have safety. It certainly elicits an immune response. How efficacious is it? Is it going to be 30% or is it going to be 90%? Your guess is as good as mine. Maybe the regulators know something, but there's nothing out there as yet, even as a preprint. So we cannot assess the safety, the efficacy of it yet. And as you see, the WHO is assessing a large number of vaccines from Pfizer, multiple AstraZeneca, different manufacturers, Sinovac, Moderna. And you see that even for Moderna, which has now been used so widely in Israel, in the US, and other places, they are estimating finishing the analysis and looking at all of their data and maybe giving approval only at the end of February. So, so they are going to take their time and be very safe because they are distributing to so many people through COVAX across the world, right? There is another view, and it would be remiss of me not to say that for the COVAX trial, which is, you might have heard the head of CSIR Institute speaking about it, that it is safe, it is immunogenic, and it is a good bet, so we send it out. But from a scientist's perspective, data, data, data. Without data, you can't make decisions. So there are two perspectives on this.
Okay. So do people who have already had COVID and recovered, do they still need to take the vaccine?
Currently, Indian government does not distinguish between the two. We don't know natural infection, how long the protection lasts. As I talked about the SARS epidemic, we seem to have T cells much later, but how good it is, if you're challenged against with SARS, will it really rescue you? We don't know because the numbers are small, we don't have that data. So the current recommendation is even if you have recovered, you should take the vaccine. Everybody has a perspective on it as well. If you have just recovered from the vaccine, say a month or two ago, you could probably let someone in the line, someone is just behind you, get ahead for a month or so ahead of you. It probably would not matter. But again, this is a decision you take in consultation with your doctor. I'm only a scientist and I'm not even doing research in this area. So with those caveats, I would say that currently, that's the guideline, but you could perhaps afford to wait a couple of months if you've just recovered.
And I have two quick questions which I seem to have missed, which are if it has been shown that the half dose plus the full dose is better than full plus full, in India, why are we still doing full plus full?
Because the actual trial, the half dose, full dose was a mistake, okay? By chance, they ended up in some small group of people giving a half and then a full. What was clinical trial design, which went through the regulators and got approval, was the full plus full. So obviously the most amount of data comes from that. I should also say that, if you're thinking 60% versus 90%, when they started this entire vaccine race to get vaccines for which will work against COVID-19, they said anything above 50% is good. Flu vaccines work only 50 to 70%, and they still protect, they still bring down the burden of disease. So, don't turn your back against it just because it says 62%. It still will have tremendous benefit. And also I want to say the Moderna and Pfizer, I think, reported well over 95% efficacy in their clinical trials. But in Israel, it is above 90, but it's suggested around 92, but with still preliminary data. So it's not a huge difference. So I think there's still tremendous benefit in taking it, even the way it is now being offered, which is in some sense, you can say that that's what you have the most data for. So that's the right dosing regime until no difference.
And why do some people get adverse reactions and not others?
Genetic heterogeneity. We are not clones of each other. And so we have variations in our immune response. And in addition, our way our immune system is trained varies. For instance, you might have heard about the hygiene hypothesis, which is that people who live in very clean countries tend to have many more allergies. And in fact, again, Shekhar Mande with Rajiv Karandikar did this analysis showing, which is going to come out, I believe, soon in Current Science, which shows that the burden, the mortality with COVID-19 might, is if you have a cleaner environment, then you seem to have a greater mortality with COVID-19. So it's not only your genetics, it's also how your immune system has been educated and your local environment. All of that make a difference. So you can't, and this is the reason why I think I said during my talk as well, that we know our populations which are susceptible, but individual outcomes is something we still do not know how to predict because we do not know that information.
Okay, so I think we will call it a day online over here. Please do go ahead with it at Prithvi. I know you're still waiting for probably what is now a cold cup of chai, but still you should have a chai for Chai and Why. Before we wrap up the online session, I just want to remind viewers of our next two sessions. 2 weeks from now, we're going to be back in the labs of TIFR with an up close with science session from a lab that investigates materials by shining light on them, the Semiconductor Optical Spectroscopy Lab. And a month from now, we'll be back at Prithvi with a stories of the unsung heroes of science, a special Women's Day session. With that, I'm going to stop the live stream. But
Arnab,
if you're at the
I
theater,
want
yeah.
to thank everybody who came to the live stream and asked really good questions. And please use the resources which are, the references which are there on the presentation, and they're all freely available, and it might, you might be able to get more information than I was able to convey here.
Yeah, but we would also like to thank Sandhya very much for taking her time to go slowly through all of this and give us the basics of how vaccines work and all the different types and all the responses that are elicited. We are looking for your response as well. With questions about the vaccine, you can always email us outreach@tifr.res.in. I'm going to put this on the chat in YouTube. I request our Facebook people to also put it on the chat. And with that, do have a nice time. Stay safe. You remember, even with vaccines, you need a mask all the time if you're going outside. So don't give up your guard just because the vaccine is around. Thank you very much for watching us. The live show, of course, continues at Prithvi.
Since we are now ending this live stream, it's nearing 12:50. Another 10 minutes, Prithvi Theatre leaves the theatre. So why not we pack off and then go out for a chai?